Our next speaker is Professor Illana Gozes. She serves as the head of the Elton Laboratory for Molecular Neuroendocrinology at Tel-Aviv University, and she is also the editor-in-chief of the Journal of Molecular Neuroscience. Today, she's here to talk to us about the remarkable drug candidate, NAP, which offers a promising avenue for the treatment and prevention of various neurological disorders. Her research is emblematic of the kind of pioneering work that can change lives and bring new hope to those affected by these challenging disorders. So let's give a warm welcome to Professor Gozes as she shares her work with us today. Professor Gozes, the floor.
Thank you very much. And please pay attention that my presentation actually runs because sometimes it is stuck. So yes. Thank you. Yes, and so I'm going to, and I'm also the president of the European Society for Neurochemistry. So this is another title that I hold. So I'm going to speak today about NAP, which we call also davunetide, CP201 and AL108. I refer to it mostly as NAP and davunetide. And I also speak about the protein where it is derived from, ADNP. So I'll speak generally on two diseases, autism and Alzheimer's disease, from bench to bedside, focusing on ADNP and NAP as a case study. So this is my second slide. I hope you see it. So we discovered ADNP about 25 years ago. And we were looking for proteins that are giving protection to brain cells against electrical blockade. So against like disruption of the synaptic function. And we discovered ADNP and we cloned it molecularly. And then we saw that if we knock ADNP completely, there is no brain. If we knock it out in an animal, in a mouse, there is no brain. So it is essential for the formation of the brain. We also saw that ADNP interacts with the chromatin remodeling proteins. So here I show you the interaction with the SWI-SNF, and this is very important for gene expression. And ADNP regulates hundreds of genes during embryonic development. Many of them are associated with synapse, and many of them are associated with the Alzheimer's disease and even schizophrenia, and also stem cell proliferation. And here what you see is that ADNP is not only found in the nucleus where it interacts with the chromatin, but also it is found in the cytoplasm, and there it is interacting with the microtubules. And I just wanted to say, like, this is, I'll go deeper into it, but amongst the genes that ADNP regulates are also genes that are important for estrogen and testosterone production, meaning that there is a dichotomy at the neurochemical level between males and females, and I will elaborate on that later. And ADNP also we showed interacts with a protein called sirtuin-1 or SIRT1, which is essential for healthy brain aging. And it interacts with it both in the nucleus at the chromatin level and also in the cytoplasm at the microtubule level. And the microtubules are actually structures that give the nerve cell the structure. Actually, without microtubules, we won't have a nerve cell structure, and we won't have axonal transport. We won't have transfer of material within the nerve cell with the long processes. When we discovered ADNP, we also saw and identified a very short fragment in it. So ADNP is a very large protein, 1,102 amino acids, and we discovered a very small fragment of it that we called NAP. This is our drug candidate that I already introduced. And this is the structure, the amino acid sequence. And NAP actually enhances ADNP binding to the microtubules, and I will explain more. And what is also known is that ADNP mutations, if they can occur in the egg or in the sperm, and then you have, when the child is born, it actually has what is called the ADNP syndrome, and the child is delayed in development within the autistic spectrum disorder and has many problems, which I will discuss later. We also discovered somatic mutations in ADNP, which are associated with the progression of Alzheimer's disease, and I will discuss this as well. So our latest finding, this is like 2023, it just came out very recently, is that NAP it can go into the cell, and it can go into the cytoplasm, but it can also go to the nucleus. And in the cell, it actually organizes the microtubules, as I said, and I'll tell you some more. And in the nucleus, it can interact with the zinc finger domain of ADNP, whereby it can actually interact with the DNA and is associated with ADNP transcriptional activity. I told you that ADNP can regulate hundreds of genes. Some of these genes, dysregulation, if we have a paucity of ADNP, are corrected by NAP treatment. And also, if we have a mutation in ADNP, what we discovered is that there may be a problem with the nuclear membrane, and ADNP can correct it. Normal ADNP and NAP can correct this problem. So why is it connected with Alzheimer's disease? How is ADNP connected with Alzheimer's disease? So in Alzheimer's disease, what we know is that that's what Dr. Alzheimer saw more than 100 years ago. There are two protein pathologies. So one protein pathology is the beta amyloid plaques, which we won't discuss, but it will be discussed tomorrow in the panel with the vaccines. We will mention it a little bit towards the end, but what the other pathology that actually develops with the disease is the tauopathy. So these are tau deposits. And what is tau? Tau is actually tubulin-associated units. So it associates with the microtubules and stabilizes the microtubule. What happens in Alzheimer's disease is the microtubules break down, tau falls off the microtubules and forms what is called neurofibrillary tangles, which progress as the disease progresses. And what you see here are the deposits of tau. If you have in a mouse half the content of ADNP, so if you don't have ADNP, there is no brain. But if you have half the content of ADNP, all of a sudden, and this is in older mice, you see these tau deposits like in Alzheimer's disease. And you also see that the enzyme that one of the enzymes that phosphorylates tau, GSK3 beta, is actually increased in activity. And then when we treat with NAP, we actually correct this. So this is one model, and we saw it in several models of ADNP mutation. So this is ADNP deficiency, haploinsufficiency. And we also saw it in human beings. So when you look at the amount of ADNP messenger RNA in the blood, they correlate actually with the amyloid deposits in the brain. But what was most striking is that when we looked at ADNP levels in the blood, they actually correlated with the intelligence test, with IQ. So meaning, you have ADNP, you actually keep and this is in elderly human beings. You have ADNP, you keep the brain alive, you keep the synapses alive, and the cognition is better. And as I said, if there is a mutation in ADNP in a child, then he will be intellectually disabled. So he will be like mild cognitive impairment in an adult. And they also, corroborating our work, a work from Taiwan showed that if you look at the entire proteins in the serum early in Alzheimer's disease, you see that the only protein that goes down in Alzheimer's patients versus controls is ADNP. And another finding that we've had is that ADNP binds to the promoter of apolipoprotein E. And apolipoprotein E4 is the major risk gene for Alzheimer's disease. It's a cholesterol carrier. And by epidemiological studies, it was found to be a major risk gene and a major risk allele, the apolipoprotein E4 for Alzheimer's disease. ADNP binds to the promoter of apolipoprotein E4, differentially regulates it in male and female mice. And another thing, again, speaking about sex differentiation, if we look at post-mortem tissues, and now we look at the hippocampus, which is one of the first areas in the brain to be degenerated in Alzheimer's disease, we actually see less ADNP in females than compared to males. This is at the level of the messenger RNA. So about NAP, and I'm a neurochemist, so I go into neurochemistry, so lots of molecules. So about NAP, it binds, as I said, to the microtubule and binding protein. So the microtubules are structures like microtubes that are essential for the structure and the passing of material in the nerve cell. And the end-binding proteins are associated with the elongation of the microtubules, but also with binding of tau to the microtubules. And NAP enhances the binding of tau to the microtubule dramatically. It enhances the binding of tau to microtubule end-binding protein into the microtubule. So like that, it can protect against tauopathy. And this is the structure of ADNP, the in silico structure of ADNP. The blue is the positive electrostatic patch, and you see here the microtubule end-binding protein binding to the NAP area. And actually, NAP enhances binding to microtubules and microtubule end-binding protein of other proteins that are also associated with autism and schizophrenia and other brain diseases. And NAP also binds, has a structure. I should say this is again an in silico modeling, and what is important for the binding of the microtubule end-binding protein is the conserved serine isoleucine proline motif. So this is the CYP motif that binds to the microtubules. Also, NAP binds to the armadillo sequence of the beta-catenin, and that's essential for the Wnt signaling, which also regulates GSK3 beta and also regulates the ACT pathway. And regulates development, and also NAP binds to SH3 motifs. And this is a structure within another protein that is associated with autism and schizophrenia that is depicted here, SHANK3. And so what I'm saying here, that NAP has many functions, and it's a very important business end in ADNP that is associated with the skeleton of the nerve cell, which is essential for synapse formation and for actually synapse activity. So that's why, and because it increases, if we have a paucity or a mutation in ADNP, it will repair because it will increase ADNP association with its partners, and it also, we assume, will correct if there are other partners that are problematic, like in SHANK3. If there is a mutation in SHANK3, NAP can repair because it will increase the residual SHANK3 that is there, interaction with ADNP and also with the cytoskeleton. So this is something to keep in mind. And so another protein that I spoke about and I wanted to elaborate a little bit is SIRT2-1. So SIRT1 is a very essential protein, and we discovered that it interacts with ADNP and that this interaction is increased by NAP treatment. And this we have shown in tissue culture, and this here it is in neuronal cells derived from induced pluripotent stem cells. We see the colocalization of ADNP with SIRT2-1 in these human cells. And we also see an increased interaction of SIRT2-1 and ADNP in the presence of NAP and also of tau. And so what we discovered in this paper is that actually SIRT2-1 has a CYP motif like ADNP. And this CYP motif is essential, as I said, for binding to microtubule end binding protein. And this is very important for maintaining this structure. And here is we have an interaction on the microtubules of ADNP and SIRT2-1 and tau. And SIRT2-1 has been shown also to protect against tauopathy. And we have shown also that it is associated with the healthy aging in centenarians. So this is connecting ADNP with a known protein that is associated with healthy aging. And what we think is that NAP, because of the enhancement of this interaction, will also protect and bring us healthier aging. And on the chromatin side, not to mix you up too much, so I said that it interacts also with ADNP, SIRT2-1 and ADNP, the genes actually interact. And what you see here is that Y11, yin-yang 1, is actually has similarities. The promoters here have similarity with ADNP, and yin-yang 1 also regulates both sirtuin-1, ADNP, and the microtubule end binding protein 1, which is also called MAPRE1. And they all regulate histone 3 modifications, as you see here. And there is also HDAC2 that is associated. And if we look, so I was quite general, but I wanted to give you this tip of the iceberg. If we look at Alzheimer's disease and we look at all these proteins that are associated with sirtuin-1, and now we show that they're actually associated with ADNP, and these are proteins that are associated at the DNA level, what we see is that there is a marked change between the normal brain, where there is a high correlation between this transcription factor and chromatin remodelers, and then in the Alzheimer's disease brain, this correlation disappears, and meaning that both ADNP and sirtuin-1 activity is essential, but there are many other proteins that are derailed in Alzheimer's disease, and this is the structure of the. This is a model of how ADNP and SIRT1 interact at the nucleus on the chromatin through WDR5, and this is the model how they interact on the microtubule, so they are essential for both compartments of the cell. And to mix you just a little bit more, there is another process that both sirtuin-1 and ADNP are associated with, and that's autophagy. This we have shown a number of years ago for ADNP. And sorry, ADNP increases the synthesis of Becklin-1, which is an initiator of the autophagy process. And ADNP also binds to microtubule-associated protein 1 light chain 3, and here we show it. So we show here the binding, and we show that there is much less binding in the haploinsufficient mice. And then we see that when we add NAP, we actually increase this binding. So, and the autophagosome is very important for taking out aggregated proteins like tau and dissipating them into amino acids, and then giving back to the cells amino acids so that the cells can live. And you see there is a connection here. So ADNP is enhancing microtubule dynamicity, as I showed you, and also the autophagy process, and by that protects against tauopathy, for example. And NAP is the key structure within ADNP, and also when added separately, that enhances this and enhances the binding. So just to show you that sirtuin-1 is associated with apoptosis and also with autophagy, and it's very central, and I think that we can draw a very similar picture for ADNP. So speaking about ADNP, it's not the end of the story. We also discovered that it can undergo mutations in the Alzheimer's brain. So this is not inherited. This occurs with aging. Apparently with aging, we accumulate mutations. And remember, Alzheimer's is a sporadic disease. And one of the proteins that we saw accumulates mutation in Alzheimer's disease is ADNP. We actually saw mutations in the hundreds and thousands of genes, but ADNP is our focus here. And here I'm kind of listing, we looked at the olfactory bulb actually, because that's an area where you have stem cell proliferation in the brain. And that's an area that undergoes, that somatic mutation can occur. And the mutations we saw, like we saw many mutations in cytoskeletal proteins. We saw mutations in proteins associated with neurodegeneration. We also saw mutations in proteins that are associated with autism and the intellectual disability. And you see here the mutations along the ADNP linear structure. And what we also saw is accumulation of mutation with the Braak stage. So, you know, in Alzheimer's disease, we can see that the Braak stage actually signifies the increase in tau depositions. So the more tau deposition we have, the more mutations in ADNP we have until it kind of plateaus. And then in an in vitro system, in cell culture, where we transfected the cells with ADNP that is mutated, and there, what we saw is a marked reduction in tau microtubule interaction. But when we add, and this is in blue, and you see the mutations below. But when we add NAP, we actually repaired it completely. So because it enlisted the residual ADNP to the microtubule and the tau to the microtubule. And there was a clinical trial a number of years ago by a company that I founded called Alon Therapeutics, looking at people with amnestic mild cognitive impairment. So that's preceding ADNP, preceding Alzheimer's disease. And we call a NAP, as I said, also davunetide or AL108. It was given intranasally twice daily or once daily. And you see that the twice daily dose actually protected the short-term memory, namely delayed match to sample. And this short-term memory is you give the patient like a picture, and then the patient has to remember this picture for 12 seconds. And what we see here is that the patients treated with twice daily, 15 milligram twice daily were faring better than the placebo treated. It was placebo-controlled double-blind, or the ones that were treated once daily with a 5 milligram dose. And so back to the ADNP syndrome, what I wanted to ask here, do the children also suffer from tauopathy? Do they also have similar findings to Alzheimer's disease? And so, as I said, these are the mutations in the children. They are not exactly the same as in the elderly Alzheimer's patients. And these are de novo mutations, so they are not inherited, but ADNP is one of the leaders in autism that are de novo mutated. So it's like a little bit like Down syndrome, that all of a sudden you have another chromosome, but here it's a mutation in a single gene that causes delayed development, motor dysfunction, moderate to severe disability, moderate to severe speech delay, autistic trait, hearing problems, gastrointestinal problem, and recurrent infection, and also early teething, facial dysmorphism, and abnormal gait. And ADNP, as I said, regulates the Wnt signaling, it regulates bone morphogenetic proteins, it regulates hundreds of proteins, as I said before. And indeed, we had the opportunity to look at a child with ADNP syndrome that passed away because of liver transplantation. There are not many cases of children with liver transplantation in the ADNP syndrome, but this is one case. And this child was 7 years old, and when we looked at his brain, we actually saw quite severe tauopathy. And here we are looking at a 30-year-old control, and we don't see any tau deposition. We saw it both by silver staining and also by anti-phosphorylated tau antibodies, and in different brain areas. This is the hippocampus, and this is the cerebellum, for example. So meaning that the children with ADNP syndrome actually undergo tauopathy, which is associated with neurodegeneration and possibly early aging. And when we looked at another model of ADNP syndrome with a mutation, so it could be both direction, ADNP syndrome, and when aging, Alzheimer's disease, with a mutation that is most prevalent in the children, it just kind of cuts the molecule almost into half, and it's heterozygous, because if you have complete knockout or complete mutation in the allele, it's probably incompatible with life. What you see here is we see tauopathy also in the mice, and very early. In 2-month-old mice, we already see tauopathy, and we repair it by NAP treatment. We also see, this is in the visual cortex, we also see a change in electrophysiology. So we see a change in a visual evoked potential. So the normal situation is here in green, and then this is in blue is the mutated situation. And then when we treat with NAP in red, we actually manage to completely reverse the situation. So we reverse the tauopathy, and we also reverse the electrical impairment in the mice. And this is just to show that indeed this is a disease of the synapse, that ADNP is a leader gene in autism, namely it's one of the leaders that is mutated, and then you have this intellectual disability, which is similar to mild cognitive impairment in the adults. And we see a difference in males and females in the areas in the brain where the reduction in dendritic spines, which are kind of the beginning of the synapse connection, we see a reduction when there is a mutation, and then we see a repair when we treat them with the NAP. And the reduction is different between males and females. Another thing that we saw is that the development of the mice is slower if they have an ADNP mutation, and they weigh less. The females, this is more in the females, and we can repair it by NAP treatment. Eye opening is slower in the female, and we can repair it by NAP treatment. And there are gait deficits that are different between males and female, and we see more gait deficits in the females. And much more gait deficits in the females. We also can measure the talking, I mean, the syntax and the vocalization in the mice. Apparently, mice do talk. And if you take away the mother, the dame from the pups, they will shout to the mother. And what you see here is the females talk more than males, like in humans. And if there is a mutation in ADNP, they talk much less. And then when we treat them with NAP, they talk the same. We actually bring them back to normality. And just because we are kind of maybe concentrating more on Alzheimer's disease, I want you to see the correlation, because in Alzheimer's disease, there is loss of speech. There is also loss of, eventually loss of motor function. And so it all, of course, and there is cognitive abilities are declining. And so it's correlating. And again, speaking about the differences in sex, if you look at the mice that have only one allele of ADNP, so they are haploinsufficient, what we see is that the males are impaired in their muscle strength, so hanging wire, grip strength, and then both sexes are impaired in object recognition, and the females are more impaired in social recognition, odor discrimination, and both sexes are impaired in social memory, and NAP repairs all the problems that I've listed here. And just speaking a little bit more, taking on the side, on the muscle, so ADNP regulates also, it is expressed in the muscle cells, regulates many genes differentially in different muscles and differentially between males and females, and this is what I'm showing you here. For example, I'm showing you here apolipoprotein E regulation in the tongue. And why is this interesting? Also because of the loss of speech in Alzheimer's disease, and also many of the ADNP cases, they don't develop speech, or they develop a very kind of very slow speech, and some of them don't talk at all. And we also looked at many muscle diseases, and we saw ADNP dysregulation. And also when we knock ADNP only in the muscle by CRISPR-Cas manipulation, we see changes in the gait, which we can repair by NAP treatment. And here is just the different diseases that we looked at, and the actual changes in ADNP. And when we look at the gait and compare gene expression between males and females to the various measures of gait, we see marked differences. So what I wanted to say here, just in a general picture, is that we see these are not obvious differences that you would look at, but there are major differences in gene expression between males and females. And ADNP regulates, as I said, testosterone and estrogen. Therefore, we should try and look at both sexes, both in animals, but also in human beings, and not mix them up. And then when we looked at, as I said, since ADNP regulates microtubules, and they are essential for the structure of the cell, and also for transport of material within the nerve cells with very, very long processes, we looked at axonal transport, which we can measure in the olfactory bulb by manganese enhanced MRI, because manganese goes in calcium channels. And what we see here is that in the male olfactory bulb, this is again mice, it's slower than in the female. You see it also here. And then if we take just the SIP motif here, just a small fragment out of NAP, that we inserted also a lysine here, so it's skip, we actually can repair that. So we can repair. And here I'm showing you also the marked gene expression regulation by the fact that ADNP is reduced, also in association with age, and also ADNP regulates the tubulin subunit differentially in males and females. So the tubulin subunit is the subunit of the microtubule. So it's not only regulating microtubule dynamic, tau binding to the microtubule, but also microtubule synthesis. So this is very important to remember because, as I said, microtubules are really essential for the nerve cell and the brain. So we also looked not only in gene expression in the hippocampus, we also looked at gene expression in the blood, in the spleen of the mice, and also at the microbiome. As you know, the microbiome is quite associated with regulation in Alzheimer's disease and also in autism. Here there is a signature of Alzheimer's disease in the intestinal microbiome. So we looked, and what we saw is, first of all, again, marked differences between males and females, also in the spleen, and also here, and this is in our model with ADNP mutation, and also what we saw is with the two models, we actually saw one microbiota strain that was similar in the two models. So we have actually in the two models, one with the haploinsufficiency and one with the tyrosine, the mutation that was done by genome editing. And so we have some similarities between the two ADNP models, many similarities actually, and that also in the microbiota signature. And this could be an objective measure to see for in terms of drug treatment because NAP corrected it. And NAP also regulated a protein called FOXO3, which is associated with autophagy, which is associated with the microbiota, and which also binds to sirtuin-1. And a major gene that the NAP and ADNP regulates is ACT1. So I will show you, and this is very important for growth and development. And here, this you saw here in the mice that have a mutation in ADNP that cuts ADNP into half, and here you see the same in the mice that don't have one allele of ADNP, so they are completely haploinsufficient. So this is a major pathway that ADNP regulates. And again, ADNP is regulated by sex hormone. This is just to remind you. So it doesn't only regulate sex hormones, but is regulated by sex hormones. And here I want to pass to human studies only. So this is not my study. This is a study published by a group in Boston about association of age at menopause and hormone therapy use with tau and beta amyloid positron emission tomography. And I think this is very, very important, because clinical trials normally take men and women and mix them as if they are identical. And I think this is a big mistake. And if you look here, you see, for example, that the tauopathy is much more prevalent in the women that had menopause early. You see here the orange line, the tauopathy versus the amyloid deposition is much higher in the women that had early menopause. So this is something to keep in mind because women are different than men. It's higher than men and higher than women that had later menopause, something to keep in mind. And then, so maybe testosterone is protective. Another thing to keep in mind is that 70% of the Alzheimer's patients are women and 30% are men. So there are more women succumbing to Alzheimer's disease and more women, and the women have higher tauopathy. And testosterone regulates GSK3 beta. So if you remember from all the details that I gave you, GSK3 beta is actually phosphorylating tau, and then tau is deposited. And this is associated also with regulation of ACT. And I showed you that ADNP regulates ACT, and NAP also regulates ACT, and they both regulate GSK3 beta, inhibiting GSK3 beta. So this all ties in together. So men are protected because of the testosterone and the GSK3 beta. And here is my working hypothesis, and you see that the NAP enhances tau through microtubule end-binding protein binding to the microtubule shaft, and then protects against tauopathy. Testosterone protects against tau hyperphosphorylation, and NAP does the same by activating the ACT pathway and activating GSK3 beta. And a reduction in estrogen will actually drive tauopathy in women. So now I want to go to the pinnacle of my talk, and this is my paper that came out in just 10 days ago, or something like that, on the 16th of October 2023. So we did a clinical trial in progressive supranuclear palsy, which is a pure tauopathy. And this, so it has similarities to Alzheimer's disease, but no amyloid plaques. It also has similarities to Parkinson's disease because there is a motor dysfunction. And we looked at several years ago, we looked at males and females together, and we didn't see an effect. But now, knowing everything that we know, we actually separated men from women. And then, of course, men and women don't weigh the same, they don't have the same height, they were the same age. But a major secondary outcome of the trial was ventricular volume. So the volume of the ventricles in the brain is actually, the larger it is, the more degeneration there is. And when we look at ventricular volume, it's not the same in males and females. So the starting point is not the same. Of course, you can't mix it if it's not the same. It's smaller in women. And then there were two, the primary outcomes were the Seidel outcome. So the Schwab and so I forgot. So it's this. Let's call it the Seidel outcome, and I'll remember in a minute. So the Seidel outcome, we looked, and that's the activities of daily living. So ADL is activities of daily living, and the PSP rating scale is actually the strength of the muscles and the muscle activity. So we will go and look approximately. So we will go and look at all of this. So if we look first at the ventricular volume, and if we look at the ventricular volume, what we see, if we look at the ventricular volume in comparison to the original volume that was there. So if we look at the original volume at the beginning of the trial was 52 weeks placebo control with about 62. The people that we analyzed, the patients that we analyzed were about 50 to 60 patients per arm. So it's placebo control, males and females, each group, about 60. So what you see here is that the placebo-treated women, if they had a larger volume at the beginning, they actually had a much larger volume in the end of the trial after 52 weeks. Namely, there was neurodegeneration in the placebo. In contrast, the treated women with davunetide or NAP, twice daily, 30 milligrams per day, there was no change. Namely, we have a neuroprotective drug. In the males, the disease is different. And in the males, we didn't see a change in both davunetide treated and placebo treated. And here it's the Schwab in England, I remember now. And here, if we look at the Schwab in England, the Seidel activities of daily living, what we see is a significant improvement. So a much slower deterioration that we already see early on with the treated women. So we see here a correlation between deterioration of activities of daily living and protection of the brain, of the ventricular volume. And if we look at the correlation in general, so we looked at other outcomes, we see that the Seidel, the Schwab in England activity of daily living actually correlates with all study outcomes in the female placebo, namely there is degeneration and then there is protection by NAP treatment. And we also see a correlation with age, and as the women are more aged, we see a maximal protection, no degeneration whatsoever with the davunetide treatment. We also see there is a geriatric depression scale. We also see protection of the females, the women in the geriatric depression scale, and we don't see that in men. And then when we compare disease progression between men and women in this pure tauopathy, the women in the measures that we looked at, the women are deteriorating much faster. So in the PSP rating scale, you see it here, and then the PSP rating scale has actually subdomains. So the bulbar, the ability to swallow, the ability to speak, you see the deterioration is much faster in the females than in the males. Similarly, in the daily activities, which are also measured in the PSP rating scale, and also the limb motor, the motor function, and here we see it also in the. This is actually the most significant. We see it in the Schwab and England, the CIDAL activities of daily living. So, and this is actually going the other direction, but this is the way that the study is calculated. So the placebo, the men deteriorates much faster than the women, meaning that you can't, and each subset of behaviors is actually deteriorating in a different level. So you can't mix it up. You have to look at men and women separately. And when you look at men and women separately for davunetide activity or for NAP activity, which I showed neuroprotection, it actually protects the PSP rating scale. It protects the limb motor, as you see here. The treated are actually deteriorating much slower, and it protects the bulbar, the ability to speak and the ability to swallow, and it protects the activity of daily living already, and it's significant and highly significant at 39 weeks of treatment. So we actually show here a correlation of a drug treatment in women only, between brain protection and actually a clinical outcome, which I think is meaningful. There is no drug for PSP, progressive supranuclear palsy, to date. And you see it here again, kind of the major findings that you saw. So these are women, and we protect the brain, we protect the activities of daily living, and the males are different. And the males they don't show this neurodegeneration, at least they didn't show it in our clinical trial. And I want to say something more about ventricular volume. So ventricular enlargement is actually a possible measure of Alzheimer's disease progression. And this is seen even before the clinical onset, meaning that one can treat before the degenerating brain and protect it before the clinical onset. So this is something to take home a lesson, and our drug, davunetide or NAP, maybe protects against it. The other thing I wanted to say, and this is relating to the vaccine story, the amyloid vaccine. So here I'm looking at aducanumab clinical trial, and what you see is actually that the vaccine maybe is dissipating the plaques, but on the other hand, there is an increase in ventricular volume. So there is neurodegeneration, and this needs to be protected. So this is something to also remember that we don't want to do, and davunetide can protect against it. So we do have some drug, and we want to start first with the children, but I would like also to go with the PSP. So we do have drug. We have orphan drug designation from the FDA and also pediatric rare disease designation from the FDA. I got the Champion of Hope in 2016 by Global Genes, and I think it's time to fulfill this hope. And I would like to thank my lab for all the fantastic work. And I also want you to stand with me with Israel, that we are under attacks by vicious people, and I hope that no vicious people will remain in this world and we will have only peace. Thank you very much.
Thank you so much, Professor Gozes. This was absolutely wonderful, really inspiring and absolutely amazing work. And thank you for joining us today from, I know what the situation is, and we appreciate even more that you took the time and are here with us today. Thank you again. So now we have some time for questions, and I see that there are already some questions in the chat. So maybe I will read them one by one. The first one is from Catalina, and she's asking, in the context of ADNP syndrome, what are the practical challenges in diagnosing and treating individuals with early onset tauopathy, and how might NAP play a role in addressing these challenges?
So in terms of the diagnosis in the ADNP syndrome is actually pretty simple. You see the mutation. So the mutation can be diagnosed very easily by sequencing, by DNA sequencing, and it's done. So this is something that can be diagnosed very easily. I would think that this tauopathy will actually affect all the children because the mutation, because ADNP is essential for microtubule maintenance and microtubule function. So I won't look precisely to see if there is tauopathy. So I wouldn't expose the children to PET scans. But what I would do is try and treat them as early as possible. And we are planning right now. I should say there is a new company that I'm working with now called Exonavis, and we are planning right now a clinical trial. So hopefully the promise will be fulfilled. And I think if we treat early enough, hopefully the results will be as we saw in the pure tauopathy PSP, and as we saw also in the early Alzheimer's patients, the amnestic mild cognitive impairment, we could bring some hope for the families and the children. And now there is a safety side effect. So what we saw in terms of side effects in the mild cognitive impairment study, there were mild side effects. I mean, there was nothing to write home about. Also but the dose was like 5 milligram once daily or 15 milligram twice daily. Also in the schizophrenia, there was not too many side effects. There was also a schizophrenia clinical trial that I didn't show because of lack of time. And actually there, the clinicians saw protection of activities, also of activities of daily living, functional activities of daily living. And something to keep in mind is that in the PSP trial, the dose was very high. It was 30 milligram twice daily. There, in men, it actually didn't improve, but there was some deterioration, meaning that the drug is doing something and you shouldn't have too much. And I think that in the men, the dose was an overdose. So this is something to take into consideration. And in the children, we will go low in the dose and actually measure to see the outcome. But there is a difference between males and females, and we will look specifically at males and females.
All right, thank you very much. So this also answers the next question. And then there is one more question. When do you anticipate the emergence of a widely available pharmaceutical drug for Alzheimer's disease in the market?
I'm working on it. So right. I'm working on it, as I said. Me and thousands of other scientists, we're working on it. I think that with the davunetide NAP, we are maybe getting at long last closer to the target.
Yeah.
Typically looking at men and women separately. Yes.
That's great. We also have a message of support from Yosef. I would like to read that out. He says, hope the best for your country, peace for all the condolences from Iranian people. We stand with you and support you.
Thank you very, very, very much. It means a lot to me.
And there is also a question from Professor Fiorenza. She's writing in our chat. Hi, Illana, interesting talk. Could you tell more on the pathway that are targeted by the davunetide drug? I can't pronounce that one.
So the pathways are, as I said, I think the major pathway is the association of tau with the microtubules and then protection against the tau hyperphosphorylation. But this is also linked with the association with the Wnt signaling pathway that is also associated with ACT and the cytoskeleton and growth in general, and also with the SH3 motif in NAP that binds to SHANK3 and other proteins that are essential also for the interaction with the actin cytoskeleton. So it's all evolving around the cytoskeleton, but I think when it goes into the nucleus and interacts with ADNP in the nucleus, it also can protect against proteins that are associated with again, with white outgrowth and with the neuroprotection, that is also important. And I forgot to say the autophagy, but it's all actually all goes into the microtubular structure, which is essential for nerve cell growth, dendritic spine growth, and synapse formation, axonal transport, and synaptic plasticity. So it all ties in together. And I've mentioned other proteins, like C2-in-1, that is also associated with the same processes. So it's like a central hub in neuroprotection. I hope I didn't mix anything up. So and it's clear.
Okay. Yes, thank you. I don't know if you can hear me.
Yes, I can.
Okay, thanks. My camera is not working. I'm sorry about this. Okay, thank you very much.
Thank you.
Thank you. All right, we still have a little bit of time for questions, so I would encourage our audience to write them in the chat if there is anything they would like to ask now. And perhaps in the meantime, I can also ask a question. So I'm sorry if I hope it's not a naive question and I didn't miss it during your talk, but you mentioned that you used NAP for patients with mild cognitive impairment. And I was curious if it also works for people who are already in advanced stages of the disease. I know that things are much trickier there and it's kind of difficult to come back from that. So I'm curious.
I think this is an excellent question, but I think I don't think that it will work in advanced stage, because what it does is kind of rejuvenating the nerve cell that is on the way to degenerate. But I think that once the degeneration already occurred, we cannot resuscitate. And I think it's not only NAP, davunetide, it's other. So there, people are thinking of cell therapy because the cell is dead. So once the cell is dead, you can't resuscitate it. What it could do is maybe recruit live cells that are there but are not in tune to do, like, to participate in a certain activity. Maybe it can do that. Maybe it can work on the residual cells that are there. And there are, like in young people, you can see if they had if the brain was traumatized, you can see that one area in the brain can take over a little bit. So, we don't regenerate, but there could be some taking over. So that's something that maybe it could do. But I would think, I would think that it would be only at the very early stages of the disease and not at the advanced stages. And also, like with the vaccines, when they did them in the late stages, nothing happened. But it's all the time people are going earlier and earlier and earlier. Once the cells are dead, that's the process. This is the process that we can't stop. So we cannot stop death. So this is something.
Yeah. Thank you for your answer. I was expecting this, but still hoping that maybe there would be some hope there, because it's also quite difficult, right, to detect it early. Like, not everyone has access to care and can get the treatment early enough. But
although maybe, like, if we think about it, if when people at a certain age start feeling that they don't remember as well, then maybe it's a good time to start taking, like, also change the activities of daily living in general, and exercise more and take care of your blood pressure and take care of your food. But if maybe some can be detected a little before the actual onset, and then drugs like davunetide will be helpful. And the other thing is actually MRI volumetric measurements, as I showed you in my presentation. So volumetric measurements actually occur before the deterioration. I mean, the reduction in ventricular volume, the increase, sorry, occurs before the neurodegeneration, before the clinical outcome. So maybe that is something that can be implemented and looked at in patients or just in the regular population, and then maybe one can treat early on. So that's the neurodegeneration precedes the actual outcome, the clinical outcome. You know, in Parkinson's disease, it is even more so that 90% of the dopaminergic cells are dead before you actually see the behavioral change. So in Alzheimer's disease, similarly, the degeneration is preceding the clinical outcome. So this could be measured, and then you can still salvage the cells that are alive and didn't die yet.
Yeah, I understand. Thank you. We have one more question, actually two more questions in the chat. Catalina is asking, can NAP or ADNP research provide insights into the broader field of neuroprotection and the development of therapies for other neurological conditions such as Parkinson's disease?
I think so. And actually, in the same paper where I described the CR2-in-1 ADNP interaction, we also looked at Parkinson postmortem brains, and we saw a similar marked dysregulation of ADNP, CR2-in-1, and all these other proteins that we looked at Alzheimer's disease. And we also had a paper where we looked at an analog of NAP, acetylated skip, and we showed that it is protective in a rotenone model for Parkinson's disease. So I think, yes, I think that this is another avenue to look at because there are similarities.
All right. And finally, a question that goes a bit in a different direction. Joanna is asking whether there is a study which involves also transcranial pulse stimulation in Alzheimer's disease.
So there are companies that are working on all sorts of magnetic stimulations. It's not exactly what I do on a daily basis, but there are companies that are working on that. And also, you know, in Parkinson's disease, there is the deep brain stimulation. So yes, there are people are looking at that as a possibility. And there were some clinical trials, but it's not there yet. But there are definitely companies that are looking at brain injury and trying to stimulate by also magnetic brain stimulation and even deep brain stimulation. I think not directly involved. Yeah. I am working with a company that does something like that, but it's remote. Yeah.
Thank you. Thank you very much. I would like to thank you again for the wonderful talk, for the discussion, and we all wish you all the best and hope the situation will finally come to a peaceful ending. Thank you again for joining us today.
Thank you. Thank you.